Back to our next lecture over viruses. This lecture is going to cover where viruses come from, how viruses are organized, how they replicate in cells, how we classify viruses, and just some overall interesting information about viruses. So let's go ahead and get started. Well, first off, viruses, and the study of viruses is called virology. The word virus comes from the Latin word virus, which means poisonous liquid. It makes sense if you think about it. They used to believe that you got sick from drinking things that were bad or eating things that were bad. So something that's poisonous, like a virus, and that's what they thought, came from a liquid. So that's where the name comes from. Now, viruses are not technically considered living organisms, and we'll get a little bit into that later. They can't reproduce by themselves. They either have DNA or RNA, not both, which is one of the classifications for cells as a living organism. So these guys just kind of lack a lot of those traits that a lot of scientists and most people would consider as a living organism. So they're not really living. They're those infectious agents that we talked about at the very beginning of this course. So these guys, viruses, are very interesting overall. In the fact that if you get a virus as a human, it's pretty unlikely that that same virus is going to be able to infect your dog. Or if your cat has a virus, that same virus is going to be able to infect you or your dog as well. So viruses are very narrow in the organisms that they can attack. They can only infect very specific organisms. So a virus that's evolved for humans, like HIV, human immunodeficiency virus, can only infect humans. So the HIV virus, it can go inside of a monkey or a squirrel or some other organism, but it can't survive there, it can't do anything. And SIV, simian immunodeficiency virus, it does the same thing in apes and monkeys, but it can't infect us or any other organisms. So viruses are very specific in the types of organisms that they can attack. Now, some organisms, some viruses, like rabies, has a very large host range, and that's what I'm referring to here is the host range, the ability that they have to infect more than one species or one species only. So something like rabies can infect warm-blooded animals. That's a lot of things. So the host range for rabies is very broad, whereas on the other hand, HIV, on the other hand, can only infect humans. So the host range for HIV as a virus is very narrow, very small. So that's what host range refers to, is the ability for viruses to infect more than one species or one species only. How many species they can infect is their host range, what host they can live in, and how many of them they can. So first off, let's talk about what a virus really is. So these are the 5 things that are used to kind of define a virus, how viruses work, what the definitive definition of a virus is. So if you follow these steps, these traits, you are a virus is kind of what this is. So a virus is an infectious obligate intracellular parasite, which means it has the ability to spread around from cell to cell, live in or on another cell. And in this case, the infectious part, and in this case, it's an obligate intracellular parasite. Which means it has to be inside of another living organism, intracellular, inside of another cell. The obligate meaning has to be part, it's obligated to. And parasite meaning that it harms the host that it lives inside. So the host cell that's been infected is going to be harmed in the process of the virus acquiring the ability to reproduce from the cell itself. So the cell will be harmed. Now, the genetic material of a virus is either going to be DNA or RNA, not both. That's one of our key characteristics here of viruses. Living cells, if you recall, have both DNA and RNA inside of them, whereas viruses are going to have one or the other, not both. And that's one of our key characteristics that makes these guys not living. They don't have both the nucleic acids, they only have one or the other. Now, viruses, I kind of like to use the analogy of war here. They don't have factories to build their own war materials, so they just go invade somebody else's country and take over their factories. And that's pretty much how a virus works in a nutshell. They don't have the ability to make new DNA, to make new RNA, to make new proteins, to make things that they need to make new viruses. So the easiest way to get that is to just take over a cell that has that stuff already. So that's essentially how a virus is going to work. They show up, they inject their genetic material, either that DNA or RNA, into a host cell. That DNA or RNA, that viral DNA, is going to reprogram the host cell and take over the host cell's genetic machinery to make proteins, to make new DNA and RNA and things. And instead of making new cellular proteins and things that the cell needs, it's now going to be making proteins, virus DNA, virus things. So the cell goes from making things that it needs to making new viruses, and that's how a virus kind of works in a nutshell. Now, once these new little virus pieces are made inside of the cell, they're going to be assembled inside of the cell as well. And then essentially what's going to happen is, via one way or another, those viruses have to get out of the cell that they're inside of. You can't go out and take over more factories and make more viruses, more cells, without being able to leave the cell that you're in. So one way or another, they have to be able to get out of the cell. And that's what we're going to talk about a little bit later in this lecture series, how they get out of the cell. And that's a big deal for different types of viruses. So is a virus alive? Now, once again, if you fall into these 5 traits, you are considered a virus. If you are a virus, you are not a living organism, at least in the overall scientific opinion. And now, life is characterized by the ability to create cellular ATP energy, respond to stimuli, grow, reproduce, and things like that, with your own species and things like that, or by your own self. If you're a self-reproducing organism like bacteria or something, asexual organism. Now, for viruses, they can do a little bit of this. They can kind of respond to stimuli. They're organized, and that's kind of about it. They can't produce ATP by themselves. They don't really grow. They don't reproduce by themselves. They have to have another species to help with this. So since they can't do some of the key characteristics of life, they're not really considered living organisms. They're just those infectious agents that we talked about earlier. They're just something that's not living, that has the ability to cause problems with inside the human body or inside other organisms. And if you think about it, toxins, poison, lots of things have ability to harm us that aren't living. So it kind of makes sense that these guys would have the ability to, since they're not alive, but they can still harm us. So let's talk about a little bit of the discovery of viruses. Viruses are one of the smallest things that microbiologists will study. That being said, they're very hard to detect since they're so small. It took a really long time, we talked about with the discovery of microscopes, to get microscopes to the point where you could even see bacteria and things underneath them. So it took even longer for people to discover viruses, to be able to see them, and let alone prove their existence, kind of thing. Be able to culture them in a lab and show that they really existed. Now, the very first person to kind of propose the idea of a disease being caused by a virus, they kind of had this idea that there was just kind of something out there that wasn't really a bacteria, but it wasn't really, it wasn't really a living thing. It was something smaller than a bacteria that they couldn't see, because they could grow bacteria really well at this point, but they couldn't grow these things. So it had to be something else that they couldn't grow, and they just didn't know what that something was. So Louis Pasteur kind of put forth the idea that this something, namely the disease rabies, they couldn't figure out what bacteria caused rabies because it was caused by a virus. They just didn't know that. They couldn't culture this bacteria ever, that maybe it was caused by something else. And what that something else was a virus, one of these small little tiny organisms, or agents, that's found out in nature, in the world, that has the ability to cause harm to living organisms, but it's not alive. Now, they didn't really understand the living part of it at this point, but they just kind of thought that there was something out there that was smaller than bacteria, and here's a name for it. And then they started looking for these things. The very first person to ever prove that these things really existed was a guy named Dmitry Ivanovsky. He's a Russian microbiologist. And he was looking for a disease called tobacco mosaic disease. And he thought it was caused originally by a bacteria. And what this disease does is it infects the leaves of tobacco plants. It makes their leaves curl up like this. They get red and yellow. It's the mosaic part, and they turn different colors, and they curl up, and then the plant will eventually lose the ability to make energy via photosynthesis from the dead leaves, and the tobacco plant will die. Now, it's passed between contact from bugs that walk on the plants, take it with them, or the tobacco plant touches another plant. It's passed through contact. And Dmitry Ivanovsky was trying to figure out what bacteria caused this particular disease. And what he did was he started using filters, tiny little filters that Louis Pasteur had developed to filter out bacteria. And he would squish up a bunch of tobacco leaves and run the juice through the filter to try to filter out the bacteria. And lo and behold, there were no bacteria that were found. So maybe he started thinking that maybe there was something that was smaller than that, and maybe it was one of these viruses that Louis Pasteur talked about. And he was able to get a hold of some of Louis Pasteur's specialized filters that he had developed for viruses to try to get these tiny little guys out. And he repeated his experiment, swishing the juices up from the tobacco leaves, running it through that tiny, tiny filter. And lo and behold, no bacteria were found, but he was able to retain the viruses this time. And he proved this by taking a sample from the juice that he filtered out with the viruses in them and scratching other leaves on healthy tobacco plants to prove that he had actually captured something in the filter that was transmissible. And that was the first proof of viruses being discovered in 1892. And then in 1901, Walter Reed, a very famous doctor, has a hospital for the military, in the military named after him, Walter Reed Memorial Hospital. Was the very first person to discover a human virus, yellow fever, spread by mosquitoes. In 1906, the variola virus, which causes smallpox. There's a couple of versions of that, variola minor, variola major, that were discovered in 1906. And then in 1933, the human influenza virus, the flu, was proven to be a virus and not, as previously thought, a bacteria. So that's kind of the little timeline of the discovery of viruses. So right around the beginning of the golden age of microbiology, they were kind of starting to get this idea of viruses in their heads. And then by 1933, people had really kind of solidified that viruses were a thing, that they were out there, and kind of really starting to hone in on what they were. So where do viruses fit in to the world of living organisms and microbiology and just things on the world? Well, we don't really know where they came from for the first part. We don't know how they showed up. We don't know where they showed up. We don't know when they originated. And you have to keep in mind that these things are extremely small. The way they replicate is very complicated. So the chances of these guys leaving fossils are just pretty much zero. So we don't know when they showed up on the planet because we just don't have fossils of them. You can't find them. They don't exist. So we don't know when they started to arise. But what we can detect is when they started to impact life on Earth. So viruses, I'll get to that in a little bit, but viruses are considered the most abundant microbes on the planet. Viruses are everywhere, inside, outside, pretty much everywhere on the planet that a living organism can be found, viruses will be there. Viruses are everywhere on and in you, and they play so many different roles inside of our bodies that we're just not aware of, that until I started studying microbiology, I didn't even have any earthly idea how important these things are to us. When a female human becomes pregnant, there's something like 26 viruses that have been dormant in her body that become active, that play a crucial role, and without these particular viruses doing this, of attaching the placenta to the uterus, the fetus will not be able to properly develop and have nutrients passed back and forth properly. So without those viruses to do that, birth as we know it would not have evolved the way it did. And that is so weird, how important viruses are to life on Earth. So those viruses aren't necessarily harming us, but they're living in and using our cells. But they do things for us that are very important, but they still harm us in certain ways. And it's just weird. So just that one little example of humans being so dependent on viruses during the process of birth, and that one specific instance, you can imagine how dependent on viruses the world is and living organisms are just across the broad spectrum of things. Viruses insert new DNA into our genes, into the genes of bacteria and fungus and plants. And there's no telling what that can do. They kill thousands of organisms. So the role that viruses have played on evolution throughout history, human history alone, is just astounding. So I could go on this forever, but I'm not. So I'm going to move on a little bit. And then the last thing here, viruses have that very primitive parasitic lifestyle. They're obligate intracellular parasites. They have to live inside of another cell. They have to use that other cell to reproduce. Oh, and by the way, when she becomes pregnant, when she's given birth, and the placenta's passed, the viruses go dormant again. So I forgot to mention that. But anyway, on to something else. So viruses. We've kind of talked about the general size of the microbiology world already. Bacteria being some of the biggest things that microbiologists will study. You can see them here. They're about the size of the E. coli. Compared to the Streptococcus next to it. E. coli is one of the largest bacteria that you look at. There's a couple Bacillus species that are significantly bigger. And then Rickettsia being one of the smallest bacteria that we will study in a microbiology lab, that you can see really well with a light microscope. And that's kind of the range of it right there. This is a yeast cell that they're all inside of a eukaryote. So, Rickettsia is about the end of a light microscope. Rocky Mountain spotted fever. E. coli is really easy to see underneath a light microscope. But viruses, all the viruses are significantly smaller. You can see down here, number 11 is a protein, hemoglobin molecule that carries oxygen inside of our blood. And then right here is the yellow fever virus spread by mosquitoes that Walter Reed identified in 1901. So it's significantly smaller than our Rickettsia. And then you can see here poliovirus, T2 phage. We'll talk about phages a little later. Adenovirus, one of the common colds. 7 here, influenza. 5, HIV, rabies, so on and so forth, up to our megaviruses, some of the world's biggest, most complex viruses. These guys are very interesting. You can see all the way down, they just get smaller as we go. So viruses are really, really small. And since they're so tiny, it's hard to see Rickettsia under a light microscope. You're not going to be able to see these guys under a light microscope at all. You just can't. So you're going to have to use one of those electron microscopes to be able to visualize the structures on the surface of a virus. They're just so small you cannot see them with a light microscope. They're just so tiny. So the largest virus is about the size of the smallest bacteria. And that's kind of the overlap of the range of these guys. Smallest bacterias, Rickettsias, are about the size of the biggest megaviruses, and that's kind of the easiest way to remember that. The biggest virus is about the size of the smallest bacteria, so pretty easy on that one. So let's talk about how viruses are put together. Now these guys are acellular, which means they do not have a cell. A meaning lacking or don't have, cellular meaning a cell. So these guys don't have a cell. They don't look like cells. They don't function like cells. They are not cells. They bear no resemblance to cells whatsoever. So just keep that in mind as we move forward. These guys are not living organisms. They are not cells. And they lack the ability to make new parts for themselves. So a virus is essentially made up of two things. You have a covering, an envelope of some sort, a capsid, some sort of covering made out of protein. And on the inside of that, you're going to have either DNA or RNA, and maybe some proteins, some enzyme in the form of enzymes. Or maybe not, it just depends on the virus species. But that's all they are. They're two things at the most basic level. A covering and a little piece of RNA or DNA on the inside. And that's all they are, protein and DNA, or RNA. And they can't make that. So if they need to make new viruses, that's the whole point of life, is to reproduce and make new versions of yourself. These guys have the same biological drive or goal, I guess. There is no really goal, it's just what they do. They're kind of computer programs. You hit A and they just continue on. But their goal, I guess, is this, for the lack of a better term, is to reproduce as well. They have to do that, obviously, to perpetuate. But they don't have the ability to make new cells. They can't, or to make new proteins, to make new RNA, to make new DNA. They can't do it. So the only way that they can do that is to hijack a living cell, which has all of the parts needed to do that already. So if I don't have a factory to make new proteins or make new DNA, and that cell right next to me does, I'm just going to take over the cell next to me and hijack his machines and make him make new copies for me. and that's how viruses reproduce. So that's pretty much a virus in a nutshell. They are a covering and a little piece of DNA or RNA inside. So let's dig a little deeper into that. So on the outside of a virus, the covering part is going to be considered the capsid. There are a couple different structures for capsids, but we'll talk about that in a second. And essentially all it is it's a nice little protective layer of protein. Nice little protected layer of protein with some DNA and or RNA on the inside of it. And that is it. The capsid is meant to protect the DNA and RNA from being damaged by the environment, to keep it in one spot, so make sure that the virus is not damaged or destroyed by the environment. You take the capsid itself, that protective layer, and the DNA or RNA inside of that. Collectively, that's known as the nucleocapsid. Nucleic acid plus capsid, the nucleocapsid. Pretty easy on that one. Now, this is the basic structure of every single virus on the planet, a capsid plus a nucleic acid. Now, some viruses can get a little fancier than this. They can, as they invade our cells, we'll talk about how they do this a little later, but essentially when they get out of our cells, when they want to leave, they're going to get up next to the cell membrane and they're going to push through the cell membrane really hard. And if you've ever put on a rubber glove and you've pulled your fingers through it really hard, you know you can pull, and eventually the rubber glove will break and all the little fingers stay on your fingers and the rest of the glove just falls apart. That's essentially the same thing that happens to these guys. They push up against the side of the cell membrane, which is made out of phospholipids. They push, and eventually they'll pop through. And as they pop through, the little ball of phospholipids that they pushed up with them balls around them and makes a little ball of phospholipids around their capsid. So they get a nice little protective bubble of phospholipids around their capsids. So if you get one of those nice little protective bubbles, not every species of virus does this. We'll talk about the differences in that and why they do this and which ones can and can't in a little bit. But if you get one of these little bubbles, you are referred to as enveloped. You have a nice little envelope of phospholipids around you. And if you don't get one of these, if you're just a capsid floating around without an envelope, you're referred to as naked. So a naked nucleocapsid or an enveloped virus is essentially the terminology that's used there. Now the capsid itself is made up of proteins that are identical little repeating structures, identical little repeating units, and identical little patterns over and over and over again. We'll talk about that in a second. And those are all called capsomeres. All the little identical structures that are put together are called capsomeres. So naked, enveloped, all these little structure here, this little pyramid is called a capsomere. This one here is a capsomere, 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 and you get the idea. So there are two really main structures for a capsid. You're going to have helical or icosahedral. And helical is essentially just a toilet paper tube with a piece of DNA or RNA wound up in the middle of it. So imagine taking a pipe cleaner, putting it into a spring shape, and sticking it inside of a toilet paper tube. That's a helical-shaped virus. You can see here each one of the little individual capsomeres, the little repeating identical protein units, look just like little Tic Tacs. And they make a little nice ring with another little ring behind it, with another little ring behind it. Just nice little stuck-on rings, kind of like lifesavers, with the nucleic acid wrapped around if it's DNA or RNA in the middle. And that's what a helical-shaped virus looks like. Very simple, repeating little capsomeres in circles all the way around, and then stacked rings like a Lifesavers candies with the DNA or RNA wrapped around in the middle. And then you have icosahedral. And if you've ever played board games that use a 20-sided die, this is a 20-sided die. They have 20 identical little triangular faces that coat or that are repeating around to make a 20-sided die shape for the virus. And that's what that is. It's got 12 corners from 20 sides, 20 different individual little faces made out of identical little repeating units of capsomeres. You can see them here. And if you zoom in on it right there in the corner, with the DNA or RNA in the middle of them. So that's the other shape, the two main shapes of viruses, helical or icosahedral. So let's go and dig a little bit more into that envelope concept. Viral envelopes, enveloped viruses are going to be mostly found in animals, viruses that infect animals. So if you recall, I mentioned that concept of host range. There are viruses out there that can infect bacteria. Those are called bacteriophages. Most of them cannot infect humans. There are viruses out there that infect warm-blooded animals that can't infect bacteria, and so on and so forth. So if you're able to invade an animal cell that doesn't have a cell wall, but still has a cell membrane. Every cell has a cell membrane, but if you have a cell wall, if you're a bacteria and you have a virus that infects that, or a plant or something, it's going to be very difficult to steal some cell membrane through that very thick cell wall. But animal cells don't have that thick cell wall, so it's pretty easy for these types of viruses to steal cell walls or cell membranes from us and other warm-blooded animals. So that's kind of what these viral enveloped viruses are going to be found mostly in. Their host range is going to be mostly found in warm-blooded animals, and that makes sense. They lack that cell wall, so it's really easy for them just to push through our cell membrane, pop off some of the protein, or pop off some of our phospholipid membrane with them, and take it with them as they go. So that's how they get their envelopes. So on the outside surface of that envelope, you're going to, let's go back just a touch. You can see them here. These are called spikes, and they stick up on the surface of of viruses that are used by the virus to stick to things to attach to the host cell. So this is what a host range is referring, or how a host range works. So viruses have different types of spikes that are found on their surface, and this is kind of like landing, like a landing zone, if you like. Human viruses have green landing zones on their spikes. Their spikes are green, if you like. This is really oversimplified, but the idea kind of works a little bit in more complicated terms, obviously. But their landing spikes are green on humans. And human cells have green landing zones. So a virus with a green landing spike can land in a green landing zone. But every other species of virus on the planet has different color spikes, so it can't land on a green zone. But a dog virus might have a yellow landing zone and a yellow landing spike, so they can land. But a yellow dog virus in us that floats past a green landing zone can't stick, and it can't attach to our cells, so therefore it can't take our cells over. This is a really oversimplified version of that, but that's really how they work. The spikes, how they stick to the cell, has to match up to the attachment point on the host cell. And if they don't, that virus won't be able to attach and infect that host cell. If they do, then the virus can attach and infect that cell. And that's what that host range concept is. If you can only infect one or two species that have a particular landing zone, or a bunch of species that have that same landing zone. And that's what that host range is really referring to. How many different species have the necessary landing zones? How many species can that virus spike attach to? And that's what that's really referring to in more of a scientific term. So what does that capsid do? What is the function of the envelope and things like that? Well, a viral envelope, or a capsid in the first place, is mostly just meant to protect the DNA and the RNA inside. A capsid protects the DNA and the RNA from harm to the environment when a virus is outside of the cell. Now, viruses have to be inside of a cell to live and reproduce, but they can also be outside of a cell, floating around in the body or in the environment. When they're in the environment or outside of the cell, they can't do anything. They're just sitting there. They can't reproduce. They can't do anything. They can't cause infection. They're just sitting there. They have to be inside of a living cell to do anything. So when they're in the environment, they have to be able to survive the environment. They can't dry out in the environment. They can't have their DNA damaged by UV light or the sun and things like that. So they have to be able to protect all of their internal structures to keep them safe, so when they get inside of a cell, they can do what they're supposed to. So that capsid really protects them from damage outside to the nucleic acid when they're outside of the cell. You don't want your DNA to get damaged, so you need to protect it. So that's what the capsid's for. Once a virus infects a cell, the DNA will leave the capsid, go into the human cell or the host cell, and then that capsid just floats away into the environment, and it is considered useless at that point, and eventually it'll be broken down by the body, or just dissolved back into the environment. So that's really what it does. The envelope, the capsid and things like that also help the virus to stick to the cell surface. They're going to assist with helping, I mentioned the spikes and things like that that help them stick. The cell membrane, if you have an envelope, it'll easily stick to another cell membrane and things like that. It attaches easier. It's also going to help with the delivery of DNA and RNA from the virus to the host cell, and we'll talk about how that happens in a little bit. It also has a handy little function of hiding you from the immune system. So the immune system just thinks it's a tiny little human cell floating around and not a virus because it's wearing a human cell coat that matches your cells. So the immune system thinks nothing of an enveloped virus, and that enveloped virus has a pretty easy time to go around and infect you. They're easier to kill, or to destroy, I should say, not kill, destroy than non-enveloped viruses, but they're a little easier at infecting you, or a little better at it. And then this is kind of the last little classification of viral structures. Anything that's not one of those two main helical or icosahedrals. And these guys are just considered complex. Anything that's not one of those two, just out of the norm. They're going to have different structures. There's ones that look like this. This is considered a phage arrangement. There's also some that look kind of like this. They have a little U in the bottom of them kind of thing, little weird shapes, horseshoes. There's all kinds of different little shaped viruses out there that are considered atypical, that are just lumped together as complex. So this is a bacteriophage, a virus that infects only bacteria. They're extremely tiny. Keep in mind, if you infect bacteria, you're going to have to be way smaller than something that infects a human cell because bacteria are so much smaller. So these guys are really, really, really tiny, and they only infect bacteria for the most part. You also have to keep in mind that bacteria have that really thick peptidoglycan cell wall. So they're going to have to be able to get through that. So that's why this guy looks a little different. But essentially right up here on the top, you can see it is an icosahedral shaped virus right there. The capsid, the little DNA or RNA inside of it, the nucleic acid, and right there is an icosahedral shaped virus. All this stuff underneath it is the complex, fancy stuff that helps this bacteriophage deliver DNA through that thick cell wall and into the virus, or into the bacteria, and we'll talk about that in a little while. So anything that's not one of the icosahedrals or the helical, still going to have that same basic shape somewhere in it. You can see that here, the icosahedral, but it's got a lot more stuff going on as well. So that's the complex shape for a virus. You can see here are, once again, our little shapes for viruses. Again, icosahedral, it's got the same main shape on top, a little more structure down below it. This is pretty much a helical bacteria with a lot of, or helical virus with a lot of proteins and stuff stuck on it. And then our different kind of normal shapes here. This is an icosahedral shape with an enveloped helical shape with an envelope. You see what they look like on the sides and things like that. So non-enveloped and things down here. Really only two main shapes. And then the last one is that complex, which is one of the two with some variation on it. And that's kind of the shapes of viruses. So viruses will fall into one of these three categories. So what is inside of that capsid? What is the viral DNA or RNA? What is the genome of a virus? Well, the genome of a virus is either, like I mentioned, going to be DNA or RNA, but never both. And that's one of the key characteristics of a virus versus a cell. Cells have both, viruses don't. They're not living. But the DNA or RNA inside of that virus is going to carry just the bare necessities that it needs to make a new virus, and that's literally it. Our cells have so much stuff inside of their DNA that's not necessary, that just kind of there, just leftover stuff from evolution, just extra DNA and things like that, that we don't really use very often or at all. But viruses, since they're so small, they don't need anything like that. They carry just the bare necessities. They carry the fewest number of genes that they possibly can to redirect the host cell to make a brand new version of the virus. And that's what they're going to have inside of them. Some viruses, like the polio virus and the really small ones down in there, carry a couple of genes, 5 or 6 maybe, all the way up to a couple of hundreds for the megaviruses and things like that. It really just depends on the size of the virus and how they infect, what the mechanism of infection is and things like that. The more simplistic the virus, the fewer the genes for the most part. So that's what the viral genome is, either DNA or RNA, with the bare minimum number of genes that that virus needs to redirect the host cell to make a brand new copy of the virus, and that's what we're going for here. So DNA-based viruses are almost always double-stranded DNA, which makes perfect sense, the same as yours, same as mine, same as pretty much every other organism on the planet. But it can also be single-stranded DNA. Single-stranded DNA viruses do exist. There are quite a few of them. It can either be in a circle or a straight line inside of that capsid. And there are also RNA-based viruses, as I mentioned. One or the other, not both. You cannot have both and be considered a virus. RNA, as it's known, is almost always universally single-stranded, but you can also find double-stranded RNA viruses. It's a little less common, but it does exist out there. There are also two different classifications for RNA-based viruses. You have positive sense RNA-based viruses and negative sense RNA-based viruses. Now, this is kind of harkening back to Biology 1, that transcription translation process with the 3 to 5, 5 to 3 concept. But I'm going to kind of simplify it a little bit and put it in the senses of English terms, or just using words here. So a positive sense RNA is something that's written, the RNA is inside of the virus is the proper direction. If you remember, things have to be 5 to 3 or 3 to 5. This happens to just be 3 to 5. It can be thrown directly into RNA translation. 5 to 3. It can be thrown directly into the ribosome and be immediately translated inside of the host cell. So this virus, its RNA reads just like this word here, reads in this right direction, left to right, like it should. And that once that host cell receives that RNA from the virus, it goes immediately into the ribosomes and immediately starts making new copies of the virus. Well, a negative sense RNA-based virus, on the other hand, has the opposite side. So if you recall, you have to have the right strain to make sense, the sense and the nonsense strand. This particular virus has the nonsense strand, the one that doesn't make sense. It's just backwards. So instead of reading left to right like normally, it reads right to left. So it reads it backwards. So when this particular virus's RNA gets inside of the host cell has to transcribe it backwards, has to reprogram it into positive sense RNA, and then it can use it. So that type of virus has to carry an extra little enzyme or an extra something with it to direct the host cell to do that extra step. So there's one more complicated step that virus has to accomplish before it can be replicated, whereas something as a single positive sense RNA virus doesn't have to do that, and DNA-based viruses don't have to do that either. So that's the little interesting thing about the negative sense RNA-based viruses. They have that little extra step to them. So inside of a virus, you may or may not have these enzymes. It depends on what you need to do. So inside of a virus, you may have a couple of extra enzymes. You may or may not, just depends on the type of virus, that are able to replicate DNA or RNA. So you, as a virus, infect the host cell, and you want to start quickly cranking out new copies of the virus. But you can't make proteins, obviously, but what you can carry are enzymes that make new DNA and RNA. So you carry those enzymes with you inside of your capsid, you inject them inside of the host cell, and then they start cranking out tons of copies of your viral DNA as well, which speeds up the process. So that's an easy little step that they can carry with them. They can also carry replicases that will copy RNA, make new copies of RNA for them. And then some of these guys, some viruses have the ability to turn their DNA or their RNA-based viruses into DNA, which is super interesting because when they do that, they can infect themselves. They can permanently embed themselves into your DNA. They're an RNA-based virus. They can turn RNA into DNA, which can be used to embed itself permanently in your genome. Very interesting process there. And that's how AIDS works. HIV, it has the ability to do that. To turn its RNA into DNA, which then can be embedded into your genome. Very strange concept. So these are some medically significant viruses, what they look like, what the common terms are, what the diseases that they cause, and things like that. Once again, don't memorize these charts. Once again, a couple more common ones here. So let's talk about how viruses replicate. Now, this is going to be for animal-based viruses first, and then we'll talk about bacteriophages. So this is going to be in cells like us, human-based viruses, warm-blooded animal-based viruses, things that don't have cell walls. So these are the 6 steps of viral replication. Now, I'm going to skip forward to the picture, and then this is the word form of what's going on here. So this is what's going to happen. Step number 1 is adsorption. Not absorption, adsorption, which means you're going to adsorb something, you're going to bring it in. Fun term there. So the bacteria itself or the host cell, the animal cell, sorry, we're not talking about bacteria virus, we're talking about human viruses, animal cells. So the animal cell is chilling out, just doing its thing. The little virus, the animal virus, usually enveloped, you can see that here, our little capsid in the middle, the envelope on the outside, is with the little corresponding spikes, is going to be floating by in the environment. The virus is going to have the corresponding spike to match up with the human cell. So once that connection is made, the spikes will bond to the cell. This is the adsorption part. The spikes stick to the host cell's membrane. Once it's stuck there, the spikes will bond, then the virus can start to carry out the rest of the steps of replication. So adsorption is number 1. You got to stick to the host cell. You do that by having the corresponding spikes that match up to the corresponding receptors on the surface of the host cell. Once you've attached to the surface, you have to get inside of that host cell. Viruses will do this a couple of different ways, but for the most part, enveloped human viruses will do it this way. They will simply just be brought into the host cell. So this is the host cell, the little virus will land on the surface of the host cell, and then the virus will just be kind of brought inside, just kind of eaten. The cell membrane of the virus is the same as the cell membrane from the human cell that it stole it from earlier, so they're going to match up. So the membrane is just brought into the cell and just kind of cleaved off there and just comes back inside. So it just kind of brings in the whole virus. So once the virus is brought inside, you have to get the virus out of the cell membrane, the envelope, so it can go to work, so it can start to do its thing. And this is called the uncoating process. So what's going to happen is the membrane is going to break down inside of the host cell. It's going to be destroyed, which is going to release the capsid from that inside of the envelope into the cell. And that's what we want. We want the capsid outside of the envelope inside of the cell. So once this process can occur, you get the capsid out, then the virus can start to invade. Now you can see here the extra steps that things that have envelopes have to do, whereas ones that don't, which we'll talk about in a second, don't have to do. We'll get into the bacteriophages, and they have a little easier time with this. So this is the big step here, coming in, getting out of that envelope, and then getting the capsid into the host cell. So once you got the capsid into the host cell, the capsid is going to be broken apart under the control of the viral genes inside of that DNA and RNA. So the capsid will be broken apart. The DNA and or RNA inside of the virus, in this case it's RNA, will start to hijack the cell itself, the human cell that it took over the host cell. It will hack the proteins, the ribosomes, and direct them to make new proteins for the virus, new capsomer parts, new viral protein spikes. It will redirect all of the machinery inside of the cell that would be making cell proteins, cell membrane, cell things, to now making viral things. So the DNA or RNA inside of the virus hijacks the cell and redirects it to do something for the virus now. So once all of those things are put together, are made, the synthesis form, they're going to be assembled, which means they're going to get up really close to the cell membrane, and they're going to form together to the capsid of the virus. So all the capsomeres will come together in the proper direction, the nucleic acid will be in the middle, and that capsid will essentially be ready to go into the world. It's pretty much a ready-to-go virus at this point. And the very last stage is the most important for a virus is to get out of that host cell. You need to leave the host cell under the release stage. So what will happen is, since it's really close to the cell membrane already, it will just push its way through, pop through, take the little bubble of the cell envelope with it, and now it will be released into the world as a brand new virus with a brand new cell envelope onto it, which can now go repeat the process on a brand new host cell. So this new virus will go do this exact same thing over and over and over again. So this is how animal viruses, human viruses, work for the most part. And this is how they cause damage to our cells. They take over our cells aren't able to do what they should. They start stealing cell membranes, which causes damage to our cells, and our cells start to die and slow down, and then our immune system comes in and just kills off the virally infected cells. So fun part there. So this is how animal viruses replicate and cause damage to human cells, as well as other cells. They steal little parts of their cell membrane. So this is where that host range concept comes into play again. I've already mentioned the spikes on the outside of the viruses. They have to match up to that receptor molecule on the outside of the host cell. So a human cell has particular virus receptors here on the cell membrane, but dogs have different ones. I mentioned this concept a lot. They're different. So the spikes that are found on a human virus won't match up to the receptors on a dog cell, or a chicken cell, or a squirrel cell, or anything other than just a human. Now, some viruses like rabies have very generic receptors, very generic spikes that hit very generic receptors on most warm-blooded animals. So they have a really wide host range, whereas something like HIV has a very narrow host range because it can only infect humans. And then you can get even narrower than that, where you have something like hepatitis, which only infects humans and only cells in the liver. So it only can find human cells that have specific liver proteins expressed on the surface that it can bond to. So host range can be something really broad like rabies, or something really narrow for something like hepatitis B that only infects the human liver. So host range really broad, really narrow. It just depends on the types of spikes that you have on the outside of your viral proteins. So once the virus has gotten to the cell, it's got to get its stuff out, its nucleic acids out of the capsid on the inside into the cell. So there's two ways that they can do this, either by endocytosis or fusion. So one of them is super easy. You literally just get up really close to the cell membrane here, and you just kind of squirt your capsid through. The viral envelope is going to just kind of do this. So we'll pretend this is the cell membrane. The envelope's just kind of do this and just kind of expand and kind of become one with the cell membrane, and the envelope that's on, or the capsid on the inside, will just kind of fall on through into the cell, and you can see that here. So the cell membrane of the cells here, our viral envelope just kind of becomes one with the cell membrane and just kind of pushes on through the virus. You can bring in the whole virus, the engulfing concept that we talked about earlier, and then the entire cell membrane is brought inside, the viral envelope breaks down, and the capsid is broken down on the inside of the cell. And then what you can have is another little fun one down here, where you have essentially the same kind of thing, but you bring it in a different form, a different way. Instead of bringing it in and breaking it down that way, you bring it in through the traditional process, and then the RNA inside the virus is going to be released through the vesicle instead of the vesicle itself being broken down. So very weird little ways that different viruses can break apart their capsids and get their DNA and or RNA inside of a host cell. So once a virus has gotten inside of your host cell, it's going to take over the DNA and RNA production of that host cell. It's going to be redirecting the ribosomes and things to make new proteins. So where does it do that, and how does it do that? Well, if you're a DNA-based virus, all the stuff inside of most animal cells that deals with DNA is going to be found inside the nucleus, and that makes sense. DNA is not found floating around inside of the cytoplasm, inside of animal cells. It would be destroyed. So if you need to be replicated in your DNA-based virus, you're going to have to migrate from the host cytoplasm into the nucleus, where you will be replicated by all the machinery that deals with that, and assembled inside the nucleus. And then you'll get out of the nucleus into the cytoplasm and then leave the cell from there. Now, if you're an RNA-based virus, you don't have to worry about that, because RNA is found inside the cytoplasm regularly. That's where it's mostly going to be transcribed into protein, but via the ribosomes. That's where they're mostly found, floating around in the cytoplasm. So the RNA-based viruses will just directly insert their RNA into the cytoplasm of the cell. The RNA will go find a ribosome and replicate there, make new proteins, make new viral capsids there, and then they will all just be assembled in the cytoplasm, make their way to the cell membrane, and leave that way. So depending on what type of virus you are, you're going to be assembled in the nucleus or just in the cytoplasm. And then once again, that positive or negative sense RNA concept comes into play again here. So there's two different ways that animal cells, or animal viruses, can leave the host. Once you're done, once you've assembled and you're really ready to go, you're right next to that cell membrane, you've got your capsid with your nucleic acid in it, ready and just fully assembled, and you're ready to go, you can leave the cell via the way that I mentioned already, which is just budding, where the cell is going to have the cell membrane expanded, the virus pushes through, and pops the little blob of cell membrane away with it, and just goes off into the cell. And this is budding, and you can see that here. The viruses are getting really close to the cell surface, pushing up through it, and eventually you can see here the little cell membrane will pinch off, taking the virus with it, and a little ball of cell membrane from the host cell. That's budding in the traditional sense. This doesn't immediately destroy the cell. The host cell that's infected can live for quite a while, like this, but unfortunately it's going to be damaged. It can't reproduce, or it can't do what it should as a cell. It's because all of its energy and resources are being directed to making viruses. So this is how they cause damage. They take over the cell, they redirect what it's supposed to do, and they make new copies of themselves. So this cell's still alive, it's just a virus factory now. And then they can leave another way. If you're a non-enveloped human virus, there's quite a few, or an animal virus, and you don't need a cell envelope. You don't have to worry about budding out of the cell. So what happens is these guys, anything that's non-enveloped or a complex-shaped virus, they reproduce this way, or they leave the cell, they release themselves from the cell this way. Enveloped viruses, budding, that's where they get their envelope from. They bud away, and that's where they get their envelope. If you don't have an envelope, you don't have to worry about it. And they just pop the cell. They do the water balloon concept. They will be assembled inside of the cell, and they can't leave. So there's a new virus here, and then another one is assembled, and then there's another virus here, and then there's another one, and eventually this poor little cell is going to be so full of viruses that it's going to pop. It's going to burst and instantaneously release all of those new viruses into the environment, or into the host body. So thousands of little viruses that are built up inside of that host cell. Eventually the cell is going to stretch and stretch and stretch. It will pop like a water balloon. The cell will immediately die. And all of those new viruses will be released. They can go infect new cells, which will eventually repeat the same process and explode and die. So when these cells pop and die, obviously whatever they were doing inside of our body is not going to be performed anymore. And that's how these type of viruses cause damage to human cells. They kill the cell, which directly causes damage to the host. So enveloped, bud, non-enveloped, lysis. They pop the cell to get out. So how do you tell if a virus has been inside of your cell if you're not infected with a virus at the moment? Viruses leave behind telltale signs that they've been there. So you may have had a virus 20 years ago and you got better, the virus shed itself, or you got over the sickness, maybe you took an antiviral medication, but the damage that that cell did, or that virus did to your cells, it can still sometimes be detected. You can see what they do to our cells. They take over our DNA, they take over our cell machinery, and things like that. So there are the things that they do, they leave behind telltale signature marks that they've been there. and each type of virus leaves behind different types of marks. So this is how I mentioned earlier that we can tell when viruses started to show up in the fossil record. We don't have fossils of them, but we have fossils of other living organisms that have viral damage to their cells. And that's how we know. That's also how we know they started to, how big of a role that they've played in evolution throughout history for different types of organisms, because we find their little telltale leftover signs everywhere in fossils throughout history. So these things are called cytopathic effects. Cyto meaning cell, pathogenic meaning danger or damage. So damage to the cells caused by viruses. So cytopathic effects. So a virus might not directly kill a cell, but it will leave behind its telltale signature as damage to that cell. And one of the things that it can do is it can cause the cell to change in size and or shape. So you can see here, this cell has combined. This is a normal human cell. It's combined itself with a bunch of other cells. So one cell has formed a giant cell. That's not normal. You shouldn't see those inside of living organisms like humans and things like that. Those multinucleated giant blob cells like that. So when you do, you know that a particular virus that causes these things to occur was there that caused that. So you can diagnose a person as having that virus, or maybe they had it at some point in time. As the virus leaves behind its little virus pieces and things inside of the cells will sometimes not be able to get rid of that viral debris. And you can sometimes see it left over inside of their cells as well as inclusion bodies. Sometimes it causes the cells to blob together and their nucleuses merge as well. This is called a multiple nucleated cell. One cell with multiple nuclei inside of it. That's not normal either. It can cause the cells to explode. Our cells don't normally do that, so if you're watching cells normally explode, or a high number of them explode, something's up. They alter our DNA. I think something like 20 some odd % of the human genome is just useless viral DNA that's been left over from countless thousands of centuries of viruses infecting us that don't do anything, that don't cause us problems anymore, but they still are there. So they've left behind their telltale marks. They're called viral fossils. Dig into that one if you want to. Very interesting concept. They can change our DNA to cause cancer. We'll get into that in a little bit. Just weird things that they can do. And then, as I mentioned, they can cause cancer directly, and those are called oncoviruses, and we'll talk about that in a second. So, once again, if you can't directly detect the virus, maybe you've gotten over the infection or things like that, or you just can't, there's no test for the virus, you can look and see what that virus has done. So pap smears and things look for these concepts underneath a microscope. They're not looking for the HPV virus itself. They're looking for the cells that the virus has gotten a hold of that has caused it to change. So HPV causes this type of stuff to occur in female cells that cause cancer, in the cervix and things like that. So that's what they're looking for when they do pap smears and things, are these cells that have been altered by an HPV virus. So they should look like normal cells, but when they aren't, they know something's up, and that's what they're looking for, those cytopathic effects. So once again, some human viruses and things like that, and the effects that they have on the cells. Please don't memorize these. You probably heard these terms before: persistent or chronic or latent. And these things are going to be used a lot for different terms for microbes: chronic states, chronic carriers, latent, and things like that. And the terms are pretty much going to remain the same for viruses and bacteria and stuff. But there's the idea is pretty much the same. But a persistent infection, where the viral cell, the host's cell, is going to be infected by a virus, and the cell is not immediately going to be lysed. You're going to be sick for a while. It's just going to be pumping out new baby viruses, or loading your cells up, or maybe not doing anything at all. And your cell's not immediately going to die. And then you have something called a chronic latent state. And this is when you, as a human or a carrier, over your entire lifetime or weeks at a time, will have an infection or sickness. So an active viral form. So the virus will be active for maybe a couple of weeks at a time. and then it'll go away, and it may never come back for your entire life, or maybe a couple of weeks later, a month later, a year later, it'll come back for a week or two, and then it goes away, and then it comes back, and it really just depends on your immune system and the virus itself, and multiple other factors. But things like measles can work like this. You can become infected with measles, and then for years you never know you've got it, and then all of a sudden you just become infected with measles, and you start having problems. Or things like herpes. Herpes simplex virus 1 and 2 cause cold sores and genital herpes. They go away for years and months at a time, and then they'll come back. People will have outbreaks, and then they'll go away again, and they come back. You may not have a bunch, you may have a few. It really just depends on the person. And then things like herpes zoster, like chickenpox. So chickenpox is a virus that most little children got prior to the vaccination. I had it when I was a kid. But chickenpox can lay hidden in your spinal cord for years. And once your immune system takes a dip, or your antibody count drops, it can reactivate itself and come back in the form of shingles. So lots of different viruses can remain hidden and not actually cause damage for years and years and years at a time before they come back and cause damage, and then they can go hidden again. And when you have one of these types of viruses, it's a chronic latent state carrier. You're in a chronic latent state where you're just waiting for the next outbreak to occur, kind of thing. Now, as I mentioned, some viruses can cause damage to the cells, and some viruses can cause cancer. So the viruses that cause cancer are called oncoviruses, and they do this kind of by overriding that biological time clock, that mitotic time clock. If you remember from biology, how cells divide. Essentially, they do this by overriding the cell's time clock and making the cell not pay attention to when it should divide, or how it should control the division process. It makes the cell ignore the checkpoints, so if the cell doesn't have the proper size or DNA or nutrients, it makes the cell ignore that, so the cell just divides. And this occurs when the virus causes damage to a cell's DNA in just the right spot. And there are cells that are viruses that are more likely to do this than other viruses, and cells that are a little more susceptible to this type of damage than other cells. And that's why combinations like HPV and cervical cancer are so common. That HPV virus is really good at causing DNA damage, and cervical cells are really susceptible to damage by that particular virus. So those two things, the cells are very prone to have damage caused in the right spot that can cause cancer and cause the cell to lose control of its growth, to cause a tumor to develop. So viruses are very strange. Oncoviruses are very dangerous if they're left unchecked. Now there's tons of different types of HPV, different strains of the HPV virus. Most of them cause warts. It's extremely easy to get HPV. You can get it by shaking hands with someone and through sexual contact and things like that. The HPV strains that cause genital warts tend to not cause cervical cancers, and the ones that cause cancers don't cause cervical warts or body warts. The warts on your hands are caused by HPV as well, and things like that. Just different versions of HPV. So that's kind of the catch-22 of HPV. The genital warts, no cancer. No genital warts, you have a risk of cancer. So that's why it's important to get vaccinated for HPV, because you don't know if you've got the one that causes cancer or not until it's too late. So let's go ahead and finish up our little bit of replication here by talking about bacteriophages, the bacteria-only viruses, the ones that infect just bacteria. These are super weird little organisms. They're everywhere on the planet that infect different species of bacteria, but they're one of the most widely studied organisms on the planet just because they're really good at using are really good at what they do. And also they're really easy to manipulate in the lab, which makes them beautiful little things to use for genetic manipulation, and I'll talk about that in just a second. Now these guys are strange. Pretty much it's the exact same replication process as a human virus or an animal virus, with a couple of exceptions. And the way that these guys get out of the cell is a little different too. So let's go ahead and dig into that. So here's the steps of replication. Pretty much the same. Words are pretty much the same. So I'm going to go ahead and talk about it this way. So this is what's going to happen with a bacteriophage. So this is how they work. I'll go back to that one in a second. So as I mentioned earlier, this is essentially just our little icosahedral head with the nucleic acid on the inside and the capsid. And down here is a bunch of fancy stuff. And this works essentially just like the little lunar lander for the moon. The little top sits up here, the little legs sit at the bottom. And as it floats around in the environment, it's going to come to a rest on the surface of the bacterial cell. And as it does, you can see here the little legs will start to twist up. As the cell lands, the legs will come down. You can kind of see that there. The legs twist up, or fold up. And as the legs fold up, all this machinery is made to allow this bacteriophage to inject its DNA inside of the bacteria. So anyway, as the little legs bend up, you can see this kind of coiled up spring here. And if you've ever had your blood taken, or you've done the little blood test at a lot of colleges and schools and things where you prick your finger with the little lancet, it's got the little coiled up spring, it's got a needle that fires out one time that hits your skin, and then it's not useful ever again. This is the exact same thing. Once the little legs go up, it triggers the little spring to fire a needle from the bacteriophage into the cell wall through the cell membrane of the bacteria. You can see that here. The little needle penetrates all the way through into the cytoplasm of the bacteria. Now, these guys don't have nucleuses or any of that stuff, so it's really easy for the RNA to go, or DNA, whatever it's inside of these guys, to go straight to work. So the little capsid will contract, squirt the DNA or RNA or whatever the nucleic acid is inside of the bacteria, and then the phage, the capsid itself, the phage body, will just remain stuck to the outside of the bacteria. And you can see that down here. This is an E. coli cell that's been underneath an electron microscope that's been highly infected with phages. And you can see them all stuck on the outside of the cells here. You can see them starting to replicate on the inside as well. So let's take a step back and talk about how these guys work once they're inside of a cell. So the bacteriophage lands. It's going to inject its DNA inside of that host cell. This is only in bacteria now, so we're talking about bacteriophages only, not human or animal viruses or things like that. So the bacteriophage lands on the cell, injects its DNA or RNA. That DNA or RNA is going to be found floating around inside of the cytoplasm of that cell. And while it's doing that, the cell itself is now going to have all of its cellular machinery, all the ribosomes and things, hijacked. It will now start cranking out new viral DNA, new viral protein structures, new viral capsids, all kinds of stuff inside of the cell, inside of that little bacteria cell. You can see them all here, new viral nucleic acid and all the little capsid parts. Eventually, all those little parts will be reassembled inside of the host cell. You can see how that process works here, what goes where, what's assembled, and how they're put together. So once they're fully assembled inside of the host cell, they are mature. And this is where that concept of the water balloon starts to come in. As more viruses are cranked out, more and more and more are made, more and more and more stress is put on the cell wall and the cell membrane of that bacteria until eventually it just can't handle the stress anymore and it pops. That bacteria will obviously die once it pops. And all the new viruses will be released into the environment where they can go land on a brand new cell and start this process over again. So this is bacterial replication, bacteriophage replication, in bacteria. Now this cycle is called the lytic cycle. This is a traditional cycle for viruses that infect bacteria, bacteriophages, to replicate and get out of the cell itself. Now, not every single cell on the planet not every single virus that infects bacteria, not every single bacteriophage works that way. Some of them have the ability to undergo this cycle here, called the lysogenic state, or the lysogenic cycle. And this is a very interesting little trick that some bacteriophages can do. Not all of them can do this, but some bacteriophages can do this. So what happens is they infect a virus. The virus infects, they infect the bacteria with their viral DNA, The viral DNA, instead of instantaneously starting to be replicated, will become embedded inside of the DNA of the bacteria itself. You can see that here. So the original bacterial DNA is going to be split. The viral DNA is going to be embedded inside of it, and then it just is now become one with the original bacterial genome. It's now embedded permanently inside of that genome. So now every single time that cell now it replicates, it's going to make a copy of its DNA. That bacteria is going to make a copy of its DNA. So now you end up with one original cell that's infected, or one original piece of DNA. Now you have two that have a virus DNA inside of it. And then those cells replicate into 4, and then to 8, and then to 16, and then to 32, and then so on and so forth until you end up with thousands of bacteria that have this copy of DNA inside of them, that have the original bacterial DNA and then a piece of viral DNA stuck inside of them. And they can do this for a really long time. They reproduce, and then all of a sudden, for whatever reason, the virus becomes active again. And instead of being dormant where it doesn't do anything, it now can become active. And now, instead of having one bacteria that's infected and you have to reinfect the whole colony, you have 1,000 bacteria that are already sleeper cells, just ready to turn on, making bacteriophage factories all across the colony of bacteria. So that's the lysogenic state. Bacteriophages that can undergo the lysogenic state can stay in that for a significantly long time, and then they'll switch from the lysogenic state instantaneously over to the lytic state and cause death and destruction inside of those bacteria cells when they choose to, or when the environment dictates that they can. Now, the other interesting thing that this can do is that sometimes this can bring some good stuff. Where it doesn't kill the bacteria. Sometimes this can add some interesting genetics to bacteria and allow them to do some really cool stuff that they might not be able to do that's sometimes good for them. And we'll get into that concept a little later in this course. So how do you grow viruses in a lab? Since they are things that require living cells to reproduce, you obviously have to give them living, growing cells to be able to reproduce. It's not like you can stick these guys like bacteria on a petri dish that's just got some juice on it or some meat juices or something, meat protein, and then they'll be perfectly fine. These guys require living cells that are actively growing and actively dividing that they can hijack and make more copies of themselves. So it makes growing these guys a little more complicated than just growing bacteria. So you have to prepare. You have to have two living organisms in this sense. You have to have the cells that are living, and then the virus that you're trying to work with anyway. So you have to take care of two things instead of just one, like the bacteria. So growing these guys is a little more complicated. So one of the ways that they grow viruses in a lab is in cell cultures or tissue cultures. And what they'll do is they will take a culture of a cell, maybe a human cell or an animal cell, and make it immortal. Now, that can be natural immortality caused by cancer. Some cancer cells never die of old age. They will divide forever, and they never naturally die. They'll die if you squish them, obviously, but they don't die of age. So those cells will last forever. Some cells we can manipulate to force them to do this. In a lab, you can't do it in a human, a whole person, without causing cancer. We haven't figured that one out yet. So you can make cells that last for a really long time, that grow forever. So when they make these types of cells, they live forever. You can grow them in a petri dish. You can just give them food and stuff on a petri dish. They can introduce the viruses to these types of cell cultures. The viruses will hijack the human cells or whatever it happens to be, the mouse cells in a petri dish, and grow and reproduce inside of that. And that's perfect to view how they cause damage to human cells, how they cause damage to animal cells, and things like that. It also is a really good way to grow them. Just a quick, easy way to do it with an easy living cell. Bird eggs. Bird eggs are kind of the perfect way to do this because that's essentially what an embryo is. An embryo is a very quickly dividing, very nutrient-rich cell. And that's what we want to grow viruses. So they take eggs that are fertilized, so usually chicken eggs, and then they will inject a virus with a little tiny, it looks like a piercing gun. They'll hold the egg up and they'll put the little gun to the side and they'll push the trigger and it'll push a little tiny needle through the surface of the egg, and it will inject the viral material into the egg embryo. And then once the embryo becomes infected, the virus starts replicating inside of that growing embryo. And then they can filter out the virus from the egg itself. So a really great way to grow viruses, very quick, very highly efficient. And this is how they grow a lot of vaccines. They grow the virus up in eggs and they harvest it that way. And that's why people that are allergic to eggs can't take certain vaccines, because they're grown using eggs. And then you have live animal inoculation. And this is necessary for studying things like SIV, where you have to actually study a living organism itself, because that virus won't grow in anything but a fully living organism. And they very rarely do these types of studies. It has to have the full organism as a whole. You can't just really infect cells on the table or an egg or something like that. That particular virus will prey on the whole body of the organism, like rabies or something like that. So you need to watch the whole animal and study how the entire system is impacted. So very rarely is this type of stuff used. It's only when it's desperately necessary. But the bird embryo is very common, as well as the cell tissues. Both of those are really common ways of growing viruses in a lab. So you can see here, these are cell culture tissues. You can see here what normal cells would look like, and then what the virally infected areas of that particular cell line would look like. It's left these big giant clear marks where the virus is, with these big giant cell, big nucleated cell here, where the virus is left behind damaged. You can study that at a plate instead of having to study that in a human. Or you can grow up viruses on a plate and then filter them out for vaccine use. And then you can see here, over here, they're injecting, now these people are doing it on a small scale with hand syringes, but the industrial scale vaccine productions use the little gun. And you can see where they inject it inside of the egg here, and then it's going to grow inside of the developing embryo. So a very interesting way to grow viruses. So why do viruses matter? Obviously, they make thousands of people sick every single year in the United States. They have billions across the world. We've obviously got a huge one going on right now. So they're one of the most common forms of, oh, I feel horrible, and then 2 or 3 days later you feel much better. Acute, quick, severe infections that come and go just about as fast as you realize they're there. You're throwing up in the bathroom for a day or two, and then you're perfectly fine the next day. Probably a virus. They also have extremely high mortality rates when compared to bacteria. Bacteria, we have antibiotics. We can deal with that. We know how to kill a living organism, how to stop living organisms for the most part. But things that aren't living, things that can float around in the air that are highly invisible, that we can't really filter out because most things are too small, very hard to control these things. So once they get inside of us, it's very hard to treat them. They're very easy to pass around from person to person, so they tend to have very high mortality rates when compared to bacterial diseases that have the same amount of diseases per year. So there's also no telling what's going to happen to people that have had the coronavirus 15, 20, 30 years from now, what the cytopathic effects of that virus leave in their body. We don't know what viruses do in the long term to people. New viruses are going to show up in the future. New viruses have been around for, they will continue to show up. They have been showing up for the past hundreds of years. So there's no telling what they do to people and our evolution in the future, because we're not there yet. So that's just interesting thought. We come to find out that viruses that we thought were harmless cause horrible effects years later that we didn't know about. And then once again, they're everywhere. They're everywhere on Earth. Every single animal, every single living organism on the planet has a virus that impacts it in some way, shape, or form. Bacteria, plants, fungus, animals, every single living organism on the planet has a virus that impacts it. And all of these ecosystems, especially in our world, are so highly interconnected that it's pretty impossible for us not to come in contact with these viruses. When we come in contact with viruses that aren't meant to be in our systems, it's the game of Russian roulette. You spin enough games, and eventually one of these viruses is going to have a mutation that allows them to go from exclusively infecting bats to now infecting humans. And when that happens, our bodies aren't evolved for that. We don't have vaccines for this. We've never studied this virus, and now we end up with things like we have now. So as the human population grows and we destroy our ecosystem and we encroach upon animals and things that carry viruses that we're not familiar with, that we don't know about, things like we're dealing with now are going to become more and more common. So that's just something of interest. How do you treat and detect animal viruses? It's a little harder than it is with bacteria. You can look for the bacteria. You can see them underneath a microscope. You can test for them. But viruses are significantly harder. You have to take a look at the overall person. What are their overall symptoms? And you can't just take a blood sample. You might have to look in the liver to find hepatitis, because you're not going to find it anywhere else. You have to look for infected cultures, infected tissues in the proper places. You have to look for parts of the virus found in the blood. You can't look for the whole virus as a whole sometimes. You can screen for antibodies instead of the virus itself, and there's really difficult to detect overall, which is why we usually just give the little quick swab. It's mostly effective, has a decent error rate for some things, and then we move on because we're mostly just looking at obviously the quick tests are much better, but for a long time they were really just diagnosed based on your overall symptoms. They're just that hard to detect that it's pretty easy to say, oh, you've got the flu. Now we have the flu test, obviously, but for a long time they just based on overall what you had. Now we've gotten into the looking at actual tissues to see how the cells are being impacted and things like that using antibodies and stuff. And the actual treatment of virals themselves using antiviral drugs is very difficult sometimes because the antiviral drugs themselves are very detrimental to the human body or the animal body, and it can cause some serious side effects. One of the drugs that's used to treat a lot of antibodies is an immunoglobulin that causes the human cells to kill themselves. And they use it to treat severe viral infections, but too much of it can cause the entire person to die. So very interesting, interferon is the word I was looking for. So very interesting use and delicate balance of antiviral drugs. And some of them can turn off human proteins and things. AZT, if you want to dig down that road, was another one of these that they used to treat HIV, and it ended up making people go blind and all kinds of other horrible side effects because of just unintended side effects. Because you have to think about the fact that a virus is only protein and DNA, and that's pretty much what we are. So there's bound to be some sort of unintended side effect that we didn't know about at the time. So let's talk about the last little bit of these guys, viruses. These guys aren't really viruses, it's just the other couple of infectious agents that are out there. Viroids, these guys are super weird. It's essentially just a tiny little piece of RNA that can cause problems in cells. It doesn't have a protein coat. It doesn't have a capsid. It's just a little single-stranded piece of RNA that might be connected to itself a little bit with some protein, with some bonds to hold the RNA into a little circle. And that's really all it is. It's just a little ball of RNA that can turn off the production of proteins and things in plants. They almost exclusively impact plants. And you can see that they cause some serious side effects in plants. So obviously this one has been infected with the viroid that causes it to grow significantly shorter. The melons are significantly smaller, different colors, loss of size here and things like that. Highly infectious between certain plant species. And you can see that these things can cause some serious damage to agricultural crops and things across the world, especially if you're trying to grow tomatoes for food. So viroids are very, very important when you're dealing with agricultural crops. You got prions. These guys are super weird. Another little infectious agent. Misfolded proteins. For whatever reason, we don't know why, a protein inside of an animal or a human's body decides to break. Now, they don't decide, it just happens. Keep in mind, they have no conscious thought here. It just happens. We don't know why. And when it misfolds, it breaks, it quits doing what it's supposed to do. And this is the weird part. When it quits doing what it's supposed to do, it tries to recruit other proteins to do that too. So essentially, this protein might be meant to hold up your brain stem or part of your cells in your brain. And then this protein becomes misfolded for whatever reason, and then it starts to try to recruit the rest of the proteins in your brain that hold things in place to leave and go do other things and not do what they're supposed to. So essentially what happens is all of the proteins in your brain that were originally meant to hold your brain in place have now turned to mush and gone and left their post. And now your brain's full of little holes and nothing's being supported, and you essentially can't function anymore because your brain's just turned into Swiss cheese. And that's mad cow disease, or Creutzfeldt-Jakob's in humans. How they get this, how these things are spread around, is once a prion spontaneously occurs inside of an organism, it will propagate itself inside of that organism and that organism alone. They live inside of nerve cells, tissue, in the nerve tissue, and brain cells. That's where these things are found. Now, when that animal is slaughtered, that meat animal, what will happen is the organism is processed for human meat. Most people don't eat the brainstem. Most people don't eat the brain itself. Most people don't eat the spinal cord. But if you do, the prions are found there. They don't get destroyed by cooking, which is kind of horrifying. They survive. They're not dead or not living. They withstand the process of being cooked. You eat that tissue and then you become infected. Now, what happened in the case of mad cow disease is they would take the spinal cords of animals, the spines, after they had cut off the ribs and things for human consumption. And then they would take the rest of that meat, the spines and stuff, and then they put it through a big giant grinder and rip off all the little tiny pieces of meat that are not fit for human consumption, that are really close to the spinal cord and things like that, for dog food and animal food and things like that, for protein, for base proteins. And what was happening is they were ripping off part of the spinal cord that had these infected cows in it. So all it took was one random cow to have this infected prion. He was accidentally or unknowingly chopped up for animal food, fed to more cows. That's what they used to do to give them base protein, feed them animal scraps. That prion survived the cooking process of making it into animal food, was processed into millions of pounds of animal food, spread out across Europe to feed more cows, and that prion infected thousands of cows across Europe in the late 90s, early 2000s because of the food practice. And that's how mad cow disease spread to humans. Humans would eat ground meat, or slaughter their own cows and things that had prions. Ground meat occasionally would have a little bit of that stuff in there, and things like that. And that's how it transmits to humans. You can also have spontaneous cases of just arising of Creutzfeldt-Jakob's disease. That's the human version of mad cow disease. Essentially just these little holes in your brain, and your brain turns to Swiss cheese. So prions are super creepy. So vaccines. Vaccines are one of those things that are really useful in getting rid or helping in the prevention of viral outbreaks. As we've seen now with the current one going on, with a lack of vaccine, how quickly viruses can spread when we have nothing to protect ourselves from them. Now, vaccines work by teaching our immune system essentially how to prepare for a viral infection before that virus is actually there. So sometimes what can happen is the virus can work by completely teaching your immune system how to prevent the infection outright, or highly lessening the infection if you do get it. So you might get it, but instead of it killing you, you might get a slight runny nose or something. And that's what vaccines are for. They're really meant to help teach the immune system to prepare for a viral infection. So vaccines have their own little world of hype around them in the public spectrum, in the sense of do they contain mercury? Well, the answer to that question is yes, they do. It's called thermosil. But mercury comes in two different forms, elemental mercury and ethyl mercury. Ethyl mercury is completely harmless. Elemental mercury will kill you. Ethyl mercury is found in the vaccines. Thermosil is a shelf stabilizer that allows them to stay stable at room temperature for a long period of time. If you eat a single-use vaccine or your doctor pulls the vaccine out of the refrigerator, it lacks thermosil, so you don't have to worry about the mercury at all if you're concerned about it, even though there's different types of mercury. And there's also more elemental mercury in a single can of Star-Kissed tuna due to our fun practice of dumping electronics into the ocean, electronic waste things, batteries and whatnot, than there is in the entire course of childhood vaccines of ethyl mercury. Do they cause autism? The answer to that question is no. I suggest you dig into the origin of that myth. It dates back to the mid-90s with a debunked doctor from England who faked his entire study. So dig into that one if you're curious. Very interesting. Vaccines overall are very, very useful. So this is how they work. There's two different types of vaccines. There's something called a weakened or attenuated or a killed vaccine. So what they do, I like to compare this to Mike Tyson. I don't want to go fight Mike Tyson. Mike Tyson would kill me. But if somebody's going to tie Mike Tyson's arms behind his back, I might be able to take him. And that's what a weakened form of a vaccine is. Essentially, they take a virus in a lab and they make it highly less infectious or highly less lethal, dangerous than it was normally, and they give it to you as an infection. So I see Mike Tyson and I walk up to Mike Tyson with his arms tied behind his back, and I can just push him over and win. So my body's already prepared to have these, prepared and kind of has an idea what to do when real Mike Tyson shows up. Or they take Mike Tyson and they put him in a blender and they give me his pieces. So they take a virus and they completely kill it, destroy it, not killed. Obviously they're not living. They just destroy the virus, completely obliterate it, and then they inject the little pieces of that virus into your body. Now, once that happens, your immune system recognizes that weak virus, that weak Mike Tyson, or all the little pieces of Mike Tyson, and goes, hey, that causes problems. We need to get prepared for that. So what they do is they make tons of antibodies, and we'll talk about how these are produced and what they do later on in this course. And they start preparing tons of antibodies to fight viruses. So these antibodies don't care if it's pieces of Mike Tyson with no arms, or full-on just Mike Tyson in his prime in the 90s ready to kill me. They don't care. They work the same way. So your body has all of these antibodies, and when real Mike Tyson, if real Mike Tyson ever shows up, they've already got fighters ready to go and take Mike Tyson out at the get-go before Mike Tyson's ever able to get close enough to me to cause problems. And that's essentially how vaccines kind of work in a nutshell. Weakened or attenuated form of the virus or a destroyed form of the virus causes your body to produce antibodies, which will then destroy the real version of the virus or prevent you from getting sick if you ever come in contact with it. So I mentioned earlier that egg concept. This is how they grow viruses up in a lab. They either grow them in cell lines or in eggs. So they grow up with lots and lots and lots of viruses in a cell propagation chamber or an egg or something like that. They infect it with a virus. The virus propagates, has tons and tons of copies made of it inside of that chamber. They then purify away the virus from the cells, cell material and things. They destroy or attenuate the virus, and then they blend the viral particles with the other stabilizers and things that trigger the immune response inside of the virus. It's submitted for approval and testing and things, and then if that's approved, it's put out into the public for use. So that's really all I've got for you guys over this lecture on viruses, how they work, how vaccines are made, and things like that. So if you have any more questions on this lecture, or you just want me to elaborate on anything at all, feel free to send me an email and let me know. Other than that, have a great rest of your day.